we're going to be talking about endocytosis. Endocytosis means inside. Exo means outside. Cytosis means cell-related. So, endocytosis means going inside, as in into a cell, and exocytosis means going out of a cell. Our main focus is going to be on endocytosis. So, we can define endocytosis by saying it is a process by which a cell uptakes material from the extracellular environment. This process requires energy and sometimes called cell drinking and cell eating. Endocytosis. The cell membrane slowly engulfs extracellular fluid and very small particles within close proximity of the extracellular fluid.
There are three main endocytotic processes. Namely, pinocytosis, phagocytosis, and receptor-mediated endocytosis. The most common and spontaneously occurring endocytotic process is known as pinocytosis. Pinocytosis takes place in almost every cell by engulfing the relatively small molecules in the extracellular fluid, otherwise known as cell drinking. During pinocytosis, the cell starts to modify its cytoskeleton structure, a process facilitated by microtubules in a cell. So basically, microtubules are small protein strings aligned one after the other to give the cell enough strength, proper structural and cell integrity, as in maintains the structure of the cell. One important thing to remember is that the reason we have a cell membrane is to protect the content of a cell.
Another endocytotic process is known as phagocytosis. It is more specified because only certain types of molecules can undergo this process. An example is the phagocyte, which is a cell found in the immune system of our body. Relatively larger food molecules and other macromolecules. This is also sometimes called cell eating. In phagocytosis, a bacteria or microtubule receptor binds on the membrane receptor. Once both the protein receptors bind together, the cell recognizes the bacteria and then gradually produces outwards and engulfs the bacteria again with the help of the rearrangement and arrangement of the microtubules. Because it is engulfed in a relatively large vesicle, it is called a phagosome. The phagosome then fuses with the limesome. which contains hydrolytic enzymes. Which are enzymes that catalyzes the hydrolysis of a chemical bond. This therefore facilitates the digestion of the material.
Now, the final type of the endocytotic process is the receptor-mediator endocytosis. This is the most specific endocytotic process. It basically involves the engulfing macromolecules, such as sugars and hormones, and of course, we also have the binding to our receptor proteins in this process. In this process, the membrane receptor proteins detect and directly engulf or go for the macromolecule. The receptor interaction signals the formation of a covering around the engulfed molecule in the vesicle. This covering is a protein layer called clathrin. When the clathrin is completely formed around the vesicle, it is known as clathrin-coated vesicle. In this case, the main purpose is to ingest the macromolecule that bind directly to the membrane receptor.
The main difference between these endocytotic processes are that pinocytosis require energy, which is quite rare in phagocytotic and receptor-mediated processes. In receptor-mediated, a protein covering is formed around the engulfed macromolecule in the vesicle, remember? This covering is called clathrin. This does not happen in pinocytosis and phagocytosis. Plus, in receptor-mediated, the macromolecule binds directly to the receptor on the cell membrane. Unlike phagocytosis, where the protein receptor binds the protein receptor on the membrane. Pinocytosis is the most common endocytotic process, and all cells undergo pinocytosis. But phagocytosis and receptor-mediated are more specific endocytotic processes.